Hello, everyone. My name is Arnesh Kumar, and my research for YRI is titled Precision at the Heart, AI Frameworks to Improve CRISPR Accuracy in Cardiac Genes. I would like to thank my mentor, Dr. Sweta MP, for their guidance throughout this project. So I will tell you about my research journey by first talking about the background of the problem, then transitioning into the computational methodology and results, and finishing with the discussion of what these findings mean for the future of cardiac medicine. So the aim of our project is to develop a novel AI-optimized framework that minimizes off-target effects in CRISPR for cardiac genes and facilitates both safe and accessible genetic treatments for heart disease. In order to achieve this aim, our main goal was to predict CRISPR-Cas9 specificity in cardiac genes that are associated with hypertrophic cardiomyopathy and understand which features play a role in influencing CRISPR precision and off-target effects. So in order to understand this urgency, we must first look at the disease. Hypertrophic cardiomyopathy is a genetic disease that causes the heart muscle to thicken, which makes it harder for the heart to pump blood. To address this disease, we can leverage CRISPR-Cas9, where CRISPR-Cas9 is a revolutionary gene editing technology that allows for making precise cuts and modifications to DNA, essentially acting like molecular scissors. However, there is a major catch of off-target effects. Sometimes these molecular scissors can cut the wrong location in the genome, and this safety risk is a clinical bottleneck that prevents therapies from reaching patients effectively. So in order to solve this problem, we started by building our high-quality data set of 5 genes related to hypertrophic cardiomyopathy, which we collected from the ClinVar database. We then selected three variants on average for each of these genes for diverse mutation locations. Following this, we used the CRISPR tool to enter a 300 base pair sequence around each mutation location for each of our genes. Using CRISPR, we were able to retrieve individual spreadsheets detailing the guides and scores for each of the gene variants, which we eventually combined into a final data set. Following through with our computational pipeline, we began by normalizing our numeric score columns, handling missing values, and performing one-hot encoding on the guide RNA sequence to allow our dataset to be readable to the machine learning model. We engineered and normalized GC content feature, which is the percentage of guanine and cytosine bases within the guide sequence. Finally, we divided our dataset into 60% for training, 20% for validation, and 20% for test, with our target variable of CFD specificity score. We ultimately chose the random forest and XGBoost models as they are capable of capturing nonlinear relationships and providing clear feature importance scores. So this table highlights the 5 core genes we selected, where we selected the core sarcomeric genes of MYBPC3, MYH7, TNNT2, and TPM1 as if directly associated with hypertrophic cardiomyopathy. We also chose LZTR1 as it is linked to Noonan syndrome, which poses a risk for early onset hypertrophic cardiomyopathy. So moving on to results, our results are mainly focused on analyzing the correlation between the key features of CFD specificity score, DOANCE16 score, and off-target count, following with the top 20 features by importance using both the random forest and XGBoost model. And our final result will be focused on the comparison for test set performance between both our machine learning models. So this is a correlation heat map matrix that reveals our key finding of a near zero correlation between the CFD specificity score and the DOANCE16 score. So the CFD specificity score indicates how well a guide avoids cutting off targets, and the DOANCE16 score indicates how efficiently a guide cuts the right location. So this emphasize the need for our AI-driven framework to maximize both safety and efficiency simultaneously, as opposed to trading one for the other. So our next result is the top 20 features by importance for our random forest model, where based on this bar plot, it indicates that off-target count was the most dominant predictor CRISPR-Cas9 specificity, which makes sense as the less places the guide could cut wrongly, the safer it is in the long run. So our next result is the top 20 features by importance using the XGBoost model, and this model was able to highlight more biological drivers of CRISPR-Cas9 specificity, where it was able to identify specific nucleotide positions, such as guanine in the 6th position of our sequence. So this highlights that our XGBoost model was able to extract cell biological signals from the sequence which the random forest model missed. So our last result here is the model comparison between our 2 machine learning models, where both models performed really well, with the random forest having an R-squared of approximately 0.93 and our XGBoost model having an R-squared value of approximately 0.92. So moving on to our discussion, we're going to be focused on the interpretation of our computational findings and their broader implications. First, focusing on how our framework compares with existing black box models through its interpretability, and second, how our computational strategy addresses clinical bottlenecks and the the ethical challenge of equitable access. So based on our results, both of our machine learning models were able to achieve high accuracy where the R squared value was greater than 0.92. And this indicates that our feature set can reliably predict off-target risk. Additionally, based on the results, we found that the GC content is not a major determinant in CRISPR-Cas9 specificity, suggesting that we should focus our feature engineering elsewhere. So the advantage that our framework offers is over deep learning models is its clinical interpretability. So this is due to deep learning models such as DeepCast13 and CNN LSTM being black boxes often. And this means that it can be difficult to understand biological reasoning behind predictions of these models. So our framework can be enhanced by combining the predictive power of these deep learning models along with the clinical interpretability of our random forest and XGBoost models. Additionally, our study is limited by being validated on a pilot data set of 15 variants, which may lead to overfitting, where our model may perform poorly on new and unseen data. So currently, gene therapies face problems with adverse off-target effects and delivery mechanisms for safe clinical use. And our research provides the capability of computationally filtering unsafe guide RNAs, as well as utilizing AI to reduce the research and development costs in laboratory experiments. By considering equitable access, we are taking the critical step of making genetic treatments equitable to diverse patient populations. So in conclusion, we were able to establish a robust AI-driven framework for predicting CRISPR-Cas9 specificity in the context of hypertrophic cardiomyopathy. In the future, we hope to expand our dataset into our full library of 119 variants spread across these 5 genes, and also validating computational predictions through investigating epigenetic features such as chromatin accessibility, as this will be the key for future CRISPR research and therapeutic applications. Thank you for listening.